Hello, everybody. Hello, welcome to the final regular session of the 2021 Delaware Estuary Science and Environmental Summit. For the last time, I'm Sarah Babulhis, Habitat Project Coordinator for Partnership for Delaware Estuary. Very grateful that you've stuck around for this final hot topic session. Should be exciting. If you have any questions or anything during the session, please reach out via the chat and our tech help people will get you an answer. Just be aware, as you heard, all the sessions will be recorded and the chat threads will be saved. All that will be available on Sched and YouTube and our website in a few weeks after the summit, so be patient. There will be 2 opportunities for questions, depending on timing during our session today. Each speaker may have a few minutes at the end of their presentation for questions, and these questions we will select via the Q&A box, so please enter your questions there. At the end of the session, there will be an open Q&A for all our speakers, and questions can be directed directly to the speakers. You can use your raise your hand feature and speak if you'd like, or you can continue to use the Q&A box. Kurt Chang from PDE will be moderating those questions. Okay, so now I'm going to throw it to our moderator for the session. Danielle Krieger can introduce herself and our first speaker. Danielle.
Thanks, Sarah. I am Danielle Krieger. I'm the Science Director at the Partnership for the Delaware Estuary. Many of you know me, and I don't want to dwell on that. So I just wanted to say that it's a tradition of the summit every 2 years to close the conference with what we call hot topics. And that's a bit of a subjective catch-all category. But if you look back over previous summits, it's really spanned everything from the Athos oil spill in our first meeting in 2005, to major climate change and watershed-wide challenges. So today, as Sarah said, we have 4 speakers, 3 15-minute presentations, and a lightning talk. And with no further ado, I would like to introduce our first speaker, Dr. David Bushek, who is director of the Haskin Shellfish Lab, and with his co-authors, Lisa Calvo and Jason Morrison, also from the Haskin Shellfish Research Lab. Dave is going to be talking to us today in Hot Topics about the status of Delaware Bay oyster resources and industries during COVID-19. Dave, you're up.
I'm unmuted, I hope, and see if I can share my screen.
You are unmuted.
All right.
And you want to switch? There you go. You're good to go, Dave.
Hang on, I'm gonna have to switch that again because it's not showing me my notes. How's that?
One more time. Yep. Good.
Okay, there we go. All right, well, thank you again, Danielle. And as she said, I'm Dave Bushek, and I do direct the Haskin Shellfish Lab at Rutgers. And I do want to make sure to acknowledge Lisa Calvo and Jason Morrison. They're both outstanding marine scientists at our laboratory. Jason focuses on the fisheries in Delaware Bay and elsewhere, and Lisa Calvo focuses on aquaculture. So to get started, I just wanted to identify that there are 3 different components that are intertwined in Delaware Bay that I'm going to talk about today. The first is the population of the oysters that you can kind of see here. Actually, this is an oyster reef in South Carolina, but they sit on the bottom of the bay in a similar way. Then we have the fishery. And of course, the fishery is dependent upon a sustainable oyster population. If it's not sustainable, then they're going to overharvest it. So we have to manage that very carefully. And then the third are the oyster farms that are distinct from the fishery, sort of in the way that if you would consider blueberry farms, maybe over in Hamilton, New Jersey, is much different harvesting those and growing those than if you were to just walk out in the woods somewhere and find a blueberry bush and harvest wild blueberries. So they're very different activities, and they're managed separately and distinctly. For a little bit of a brief background then, I think everybody knows that oysters exist pretty much throughout Delaware Bay. The highest abundance is up here in the upper half of the bay. but they also exist in the tributaries in the bay and in the marshes that surround the bay as well. Salinity is probably the dominant factor controlling their distribution and their abundance, affects recruitment, growth, condition. All those increase with salinity, and so does predation and mortality. So you have higher survival up here in the upper part of the bay, and as you move down the bay, you get greater and greater mortality. The fishery operates on these natural beds that are in the upper part of the bay, up here. And the oyster farms exist down in the lower part of the bay on what were formerly leased grounds predominantly, and then there's a new area of intertidal oyster aquaculture down here. And those are both shown. So here's an oyster fishing boat, and here's an oyster farm. All right, so starting with the population, one of the primary functions of our laboratory is to provide an independent assessment of the oyster abundance in the fishery. And we've been doing this since 1953. We have a highly accurate process that's used by the resource managers in the state of New Jersey and the oyster industry to arrive at a sustainable quota each year. Without going into too much detail, what we do is we go out on an industry boat here, and we sample the fishing grounds. And we like to use an industry boat because they understand what we're sampling, and also we're sampling what they're fishing, as opposed to if we're using a scientific boat or research vessel. Our staff then processes some of that on board and brings that back and does some further analyses and enters that into some computer programs, which ultimately helps us calculate the density of oysters on the bottom of the bay, which we can then scale up to the entire population. And so here's sort of a way that we scale that up. It shows that the fishery has several regions that are managed differently. This pie chart simply shows the acreage of the different areas, indicating that this area down here has by far the highest amount of acreage, but not necessarily the most oysters, because the density of oysters increases as you go up bay. This area management helps us account for different population dynamics, whether there's high rates or low rates of recruitment, whether mortality is high or low. It also helps us spread the harvest across the bed. So whatever quota is set for this area can't be harvested for any other area. So that helps ensure that overharvesting doesn't happen in one area. And then this assessment of the population is then formally reviewed and by an external committee that helps us develop harvest and management recommendations. So one of the last meetings that we were able to host in person at the Haskin Lab was the 22nd annual stock assessment in February 2020, just over a year ago. And our purpose of that is to determine the status of the fish population and recommend a quota that's sustainable. Back in that a year ago, they concluded that the fishery was being managed sustainably, and therefore a quota could be harvested. And the size of that quota is shown here, 97,000 bushels, with another 17,500 added post any enhancement activities. So the enhancement activities are part of the sustainability for the fishery. So what they're trying to do there is increase the abundance of oysters in the bay. When they increase the abundance of the oysters in the bay, we don't increase the percentage that can come out, but 2% of 100 is not as much as 2% of 1,000. So if you can get the population to grow, the actual abundance that you harvest increases. Okay, so let me see, what did I miss here? Oh, and so just to give you an idea of the potential value of this in the fishery, this amount of harvest would equate to about a $6 million dockside value. After they put that through a packing house and box it up to take it out to restaurants or retail markets, it might increase in value to about $14 million. And then if those were to sell all those oysters, say at a raw bar for $2 each, that would be a $61 million value. And so those would be annual values for this size of a harvest. But as we all know, things, well, they were looking bright. There was a dark cloud on the horizon. And as soon as this executive order was put in place, restaurants were closed, orders were canceled. This actually resulted in some places shipping product back, maybe that had already arrived or that was in transit. I don't know what the guys were expecting they were going to do with that. All of a sudden, they shipped out truckloads of oysters, and here they are arriving back at their door. On top of that, state employees were furloughed. That stalled a lot of the fisheries management activities that are critical to the sustainability of the fishery and also maintaining that allowable quota. So ultimately, this resulted in the quota dropping by 15% because that extra additional quota back here, this 17,500 bushels had to be pulled out without those activities happening. So what are the oystermen supposed to do if they don't have a market? Well, a number of them would just decided they would simply wait out to pick back up and focus on maintenance and repairs. And you might remember that there were claims that COVID was under control and would probably go away once temperatures warmed in April. So they just have to sit this out for a month or two, and then they could get back to harvesting. So that was the plan for some of them. Other oystermen still have leases down on these private grounds where they can plant oysters. So they were able to continue harvesting and move those oysters down to their private leases. The intent there is that later when the markets open up, they can harvest them. And they just have to hope that they don't die from disease or predation or siltation in the meantime. But of course, that all costs money, either one of those things, maintenance and repairs or moving your oysters. So the CARES Act and programs such as PPP really were effective in helping a number of the companies allowing them to continue to harvest or continue to repair their vessels and things, even though they didn't have sales to come in and supplement those activities. So that was really a good thing that happened for them. And in the end, though, while the initial harvest rate had dropped off by 75%, by the end of the season, most of their quota had actually been harvested. And so that brought us to last month when we were able to have the current stock assessment. And this stock assessment was a little bit different because until July, we really weren't able to get out and do any of the monitoring that we usually do every month with the state. But from July on, we were able to pick up all our monitoring programs and conduct our assessment in the fall of sampling the fishery. And so we successfully held the 23rd Delaware Bay Oyster Stock Assessment the same way we're doing this meeting, over Zoom. So that was a little bit different adventure for us as well. But what I'd like to do is show you a little summary graphic here that we use to get an idea of what's happening with the population. This is called a target and threshold plot, and it's used to help us make management decisions. On the bottom, on the x-axis, you see the abundance of oysters of all sizes. And we have a target abundance that we want the population to remain above of about 2.3 billion oysters. And then over on the y-axis on the left, we have the abundance of market oysters. Those are the oysters that can be harvested. And that target abundance is around 400 million oysters. And so if we maintain those abundances up above those levels in this green area, if you will, then the population is doing pretty well. And what you see here, these points here show where the population has been over the last 4 years. There was a little bit of a decline this last year, but in general, we're staying in this green area. And so that's good. So staying in this green area is an indication that we're managing the fishery in a sustainable way. And so the result of that was that 2021 quota is going to be about 114,000, almost 115,000 bushels. And this was actually approved yesterday afternoon at the Shellfish Council meeting. If you're interested in the details of the fishery and how we manage it, you can get those off our website shown here. So what about the oyster farmers? They really weren't any better off. The market shutdown following stay-at-home order produced the same problem with them, with orders being canceled. I don't think they had as much product shipped back necessarily, but the farms had to still be maintained. Those oysters continue to grow. The cages and the bags that they're held in continue to get fouled up. So they still had to send employees out to maintain those animals or they die. So for them, again, the CARES Act and the programs that were related to it became pretty essential for them to continue operating. But some of them had learned that they could pivot to direct sales directly to consumers. And other ones later on found out that there's an opportunity to provide oysters for restoration. And I'm going to speak about these two options next. So here's an example of direct sales market here. So basically what they did is they changed their packaging to make it into small retail packaging, so it could be sold directly to consumers. They could either come and pick it up from them, or they could go to a market, or they could actually have it FedEx shipped to their door in some cases. With it, they provided shucking kits and instructions on how to open oysters. Sometimes they had YouTube videos to send along, or even did Zoom sessions, some of them did. And some of them included recipes on how to cook them if people didn't want to eat them raw, as shown here in the picture. So while the initial price and sales volume had plummeted, it slowly recovered. And the recovery towards over this past fall and winter was actually pretty strong. And last month, one farmer told me that demand had outpaced supply as Valentine's Day approached, probably an indication that a year in quarantine requires a little bit extra stimulation for some romance around Valentine's Day. All right. So one of the other options was oyster restoration. So there were still many oysters on their farms that had grown out of market size. There's a specific size that the market really prefers, consumers prefer. You don't want to try and shuck a 5-inch oyster and swallow it whole. So they like smaller oysters, 2.5, 3-inch oysters. But these oysters continue to grow if they weren't marketed. And during the summer, when we're shut down, they grow really fast. So those were taking up space on the farms. They had to be maintained. Offering them up for restoration became a pretty viable option for organizations, for a lot of these farms. But most of the organizations that wanted these recognized that, you know, this is a business that these oyster farmers are growing their oysters from. They can't just afford to give away half their crop.
2 minutes, Dave, 2 minutes.
Okay, I'm just about done. And so 2 organizations stepped up to do this. The first one was organized by Lisa Calvo at our lab with New Jersey Sea Grant through their COVID Rapid Response Program. And the second was a multi-state effort funded by TNC and Pew called SOAR. SOAR stands for Supporting Oyster Aquaculture and Restoration. So both these programs sought to provide oyster farmers with financial relief while enhancing restoration projects, and the others, and the oysters that suddenly had no market would become broodstock for the next spawning season on the restoration projects. It's sort of a win-win, if you will. Rutgers and Stockton researchers helped and assisted by collecting data. You can see that down here. The NJDEP, you see some NJDEP staff here that were helping to coordinate this so that it all happened within all the policies that are there. And in total, there were 27 farms that planted 688,000 oysters on 4 restoration sites. So it's quite a success story and sets up a new avenue of sales and relationships between oyster farming and oyster restoration. So while COVID-19 presented several challenges, I think we can agree that Delaware Bay oysters farmers and fishermen are undeniably resilient. And I guess I'd be remiss if I didn't end by reminding you that oysters are good for you and good for the environment every day of the week. So thanks for your attention, and thanks to PDE for letting me provide this update.
Great, thank you so much, Dave. I think we're probably needing to keep on schedule. So if we could hold questions to the end of the session, that would be great. And again, reminder to put your questions into the Q&A so Dave can maybe start preparing on responses if you have any for him. So our second talk in the hot topics session, I'm delighted to introduce Adrianna Zito-Livingston from TNC in New Jersey with co-authors Doug Fairclough and Mattia Jepsen and Laura Crane. And Adrianna is going to be talking about tracking vegetation response to beneficial use of dredged sediment for high platform marsh restoration at 3 sites in southern New Jersey. So Adrianna, the mic is.
All right, can you guys see my screen?
Yes, free to present, do 9%.
Yep. All right, can you see the right screen or do I need to swap?
Nope. swap.
Well,
Such a tradition. Thank you, guys. And now for something completely different. That was a really good presentation, Dave. I really appreciate the opportunity to speak during Hot Topics. I work for the Nature Conservancy here in New Jersey, and it's been my pleasure for the last 4 or 5 years to work on a beneficial use series of beneficial use restoration projects that are led by New Jersey DEP. And our responsibility, we've been engaged in these pilot projects. There are 3 of them in South Jersey. We've been engaged with them from the beginning to ensure that a comprehensive monitoring approach was developed. And then it's been our pleasure to continue measuring vegetation response over time. This was not done alone. So the Conservancy has been working with DEP. They're the primary, they're the lead, they're the landowner. Oops, sorry, just bear with me. So this project was funded. So up in the right-hand corner, we have funders. So the project was funded through the Department of Interior Hurricane Sandy Recovery Fund, administered by NIFWIF, and the grant was received, it was awarded by DEP, to DEP, sorry, and continued funding. So that covered implementation and initial monitoring, and the EPA has been generous enough to continue support continued monitoring at this site. So there's not much you can figure out in 2 years, and we appreciate the opportunity to continue a longer time series in tracking the recovery of these sites. There are a number of other monitoring partners. This is a really big effort, and I'm just gonna tell you a small piece of the puzzle today. I hope I whet your appetite. So beneficial use of dredged material was piloted at 3 different locations in New Jersey. It hadn't been tried here before, and the Sandy funds provided an opportunity to give it a try. These projects were implemented between summer of 2014 and spring of 2016. And while several different features were incorporated into beneficial use, that's a kind of a catch-all term, including a dune and beach construction and elevated nesting habitat that I know Lenore Tedesco has presented on in this meeting and others. I'll be focusing today on the marsh restoration, marsh platform restoration piece of beneficial use. I'm also going to be fairly brief in my description of the different sites. They've been presented at previous summits, posters, and also within this meeting. So as I said, I'm just presenting a small piece of the puzzle. There are 3 different project locations. You can see them on the map on the left. Sediment addition occurred concurrently with dredging of the intercoastal waterway on the 2 Atlantic coast sites, Avalon and Ring Islands, working with the Army Corps of Engineers. And then Fortescue Creek was the dredging project that supplied sediment for Fortescue, and that was in cooperation with the Department of Transportation. 2 screens seems like a really good idea until you're trying to drive a mouse between them. Bear with me. I wanted to put a plug in for a pre-recorded talk that also dives a little deeper into elevations. So definitely check out Brittany Wilburn's pre-recorded talk. It's also, I believe, linked at the bottom of this session. So as I said, there's a comprehensive monitoring program. This actually is a laundry list of some of the metrics that are being monitored. As I said, I'll focus on what the Nature Conservancy has been monitoring over the last 5 years, and that's vegetation. We've also been measuring bearing capacity, monitoring sets, and surveying macroinvertebrates, but there's no time for that today. So the overall project goals for all 3 beneficial use projects were to increase and then maintain elevation that provides ideal tidal flooding and flushing for native salt marsh species. The second goal is to increase robust native vegetation. And the third goal was to return to baseline for conditions for all other metrics, unless they were expected to decline due to habitat conversion. So it's a really fancy way of saying we wanted to do no harm, unless we intended to say, you know, convert a habitat like a pool into marsh platform. So there were some intentional choices and goals related to these projects that involved habitat conversion. But aside from that, we wanted to at least have conditions return to baseline. So there's a number of, when we're looking at vegetation monitoring and metrics, there were 3 ways to really look at, actually 4 different ways listed here of ways that we looked at vegetation metrics and to be able to look at increasing response. So kind of a bare minimum assessment is the number of plots. We did all of our monitoring in permanent vegetation plots, actually, I should say. You can see here on this meter squared quadrat. We used a before after control impact experimental design. So there are control sites associated with all of these sites. The benefit of this approach is that in a heterogeneous landscape, we're able to track the succession of vegetation at a specific place and time. So lots of things are changing, and we can have a really nice idea of what's happening at that meter squared level if we use permanent monitoring plots. The downside is some of these sites are really large. And so kind of trying to study, in some senses, studying a postage stamp to understand what's happening on a football field. Oops. Some real driving issues. All right. So at first, we have the proportion of total plots and the number of those plots that become vegetated over time as our first signal of response and recovery. So you can see in the bottom left-hand corner examples of 3 basic habitat types that we characterized in the field really rapidly. So if a plot had more than half of its area covered with vegetation, or vegetation was present in more than half of the plot, it was considered vegetated. If more than half of the plot was bare, it was considered bare. And if more than half of the plot was considered is a pool, then it was considered a pool plot. So a really coarse assessment. We also, within that meter squared plot, looked at percent cover, and that's a canopy cover. And we used burnt blocklet cover classes to assess cover. We also measured stem heights. I'm not going to share those data today in the interest of time. And we looked at the variety of species present. So diving right into Fortescue, this is an image where you can see a close-up of the almost 7 acres that were restored here. Those 2 great green blobs are the areas that received placement. There are control sites down here and then also up here for comparison. So the average depth of placement here was around 7 inches. It varied from 0 to 18 inches. The materials used were a mix of silt and sand. So diving right into these signals of veg response, looking at the habitat types, as I said, this is the earliest signal of how vegetation is reestablishing. So it's probably no surprise to anyone that when you add a significant amount of material to the surface of a marsh, you go from a somewhat vegetated and stressed marsh to a marsh where there's a lot of bare ground. And so we see that signal here. So in year 1, there was a lot of bare ground, and it's been a slow creep back to a vegetated area. You can see the control plots here on the left, placement plots here on the right, and this is just a percentage of plots on the y-axis. And I should say the letters at the top signify statistical similarity. So we can see there was really no change in the proportion of vegetated plots at the control site over time, whereas there were significant differences. You know, we have the precondition where it was mostly vegetated. And again, this is a really coarse assessment. We're not saying these plots were healthy. They just had vegetation in them. This is a measure of vegetated extent. Over time, then we had mostly bare plots. So that's a significant difference. And encouragingly, by year 3, a large proportion of our plots were vegetated plots again. And that trend has held. These sites were last monitored in 2019, and DEP is planning to do monitoring again this summer in this growing season. So we're hoping to be able to track this trend over time. So it's really encouraging that at this site within 3 years, we had pretty good recovery of the vegetated extent. The other measure, the more traditional measure of vegetation recovery is the percent of vegetation cover within a meter squared plot. You can see, looks like my letters are gone, but these dots are pretty close together. So in years since placement, after 4 years of monitoring, of annual monitoring, we're now seeing the placement plots being relatively or pretty darn close to the percent cover present in our control plots, and that's really encouraging. And if the stats were on here, you would also see this is very similar to the baseline condition. So we've done it. The percent cover of vegetation at Fortescue is at this moment equal to that of the control sites and equal to post placement. But we want to do better than that. So it's a good thing that we're going to continue monitoring. So hopefully this trend will hold and we'll see uplift, which is ultimately the goal. So this is just an encouraging image. This is not necessarily representative of every plot, although at Fortescue it is pretty typical. You see the transition from bare ground in a bare plot to a plot that you can see vegetation is going to come in, then a vegetated plot as of 3 years, and then a pretty densely vegetated plot. And that percent cover increase after 4 years. We also look at the dominant species. And so as we're tracking how vegetation is recovering here, we're really seeing succession happen. So we have our early colonizing species come in right after placement. We see some plots that are dominated by Disticulus, and at other sites we've had Salicornia as an early colonizer. And then over time, we see a transition to Spartina alterniflora. And Fortescue, the hope was that, the design was to create high marsh. So really looking for Spartina patens. And you can see as we get into years 3 and 4, we did have plots start to pop in and increase of plots that were dominated by Spartina patens, so that target species, which is really encouraging as well. So one of the opportunities we had with the time series of data and the other monitoring aspects that were being monitored on the site simultaneously is to be able to start to tease out the why that vegetation recovery was happening. This is really important for designing future projects. So understanding not just that you can do this and it'll work out okay, but being able to craft a project where you can optimize success and increase veg cover and really help with performance over time. And so we did a series of multi-metric analyses looking at what those causes were. So at Fortescue, there was a positive relationship. You were likely to see veg cover more in the interior of the marsh when you were further from a water body.
2 minutes, Adrianna. 2 minutes.
Okay, thanks. And then also with vegetated distance. So the more likely you're more likely to be vegetated if you are immediately adjacent to a plot that is also vegetated. So I'll just summarize for Fortescue, the highlights were the number of vegetated plots and the percent vegetated cover are both back to baseline condition. And we're really encouraged that over time, we had a really great growing season last year, even though we weren't monitoring, that we'll be able to detect that change this year. This Fortescue, it's good that I only had time to highlight Fortescue, had the largest increase in percent vegetated cover of all the sites. And one of the leading hypotheses that we have as far as why that happens, and it was supported by the multi-metric analysis, is Fortescue has a lot of edge relative to the total size of the site. And so there are lots of places for vegetation to come in. Don't panic or get seasick. Okay. Great. So the highlights at Avalon and Ring Island, I'll just briefly cover. Avalon is also recovering. It's a much larger site. And so our ability to detect vegetation recovery, and we are seeing indications of those percent of the permanent monitoring plots that have vegetation in them, that they are increasing. It's just a slower signal. These areas were very large and they were treated with silt. And so it took longer for them to stabilize and understandably longer for them to vegetate. DEP has been looking across these sites with drone imagery, and we can see vegetation really coming into these sites at a rate that's much higher than we're able to detect in our permanent monitoring plots. Right, I will wrap there because Sarah threatened with a hook. But I am excited to answer questions about these projects and to continue to engage and share what we know to support further and future projects.
Awesome. Thank you so much, Adrianna. We will continue to hold Q&A until the end, but I actually don't see any questions coming in. So there's over 100 people on this webinar. Please, again, put any questions you have for any of the presenters in the Q&A. And at the end, we'll take a few of those questions. So moving on to our next third talk in hot topics, Alex Parker from Cal State University Maritime Academy with co-authors Joanna York, Margaret Dolan, and David Walsh Alex is going to be speaking, pretty intriguing title, I think. Does ammonium supplied from municipal wastewater suppress primary production in the urban Delaware River? So Alex, good to go?
Great. Hopefully everyone can see my presentation and perhaps you can see me as well and hear me. So I'm thrilled to have the opportunity to share with you results from a study that we've been working on to look at this kind of counterintuitive question, which is, is there a role for anthropogenic nutrients, specifically ammonium, in actually suppressing rates of primary production in the Delaware? Before I get into my talk, I did want to just recognize my co-authors, including Dr. Joanna York from the University of Delaware, who's the co-investigator on this project, Maggie Dolan, research technician, and Dave Walsh from the Woods Hole Group. This work was funded by the Philadelphia Water Department, and they've been excellent partners with us, both in the sort of formulation of the project, as well as along the way with ideas about how to do this study well. So I did put up this kind of provocative title. It actually doesn't belong to me. This idea was first proposed for the Delaware estuary in the early 2000s by Koei Yoshiyama and John Sharp, in a paper in Limnology and Oceanography. And they looked at a 25-year data record of biogeochemistry throughout the Delaware estuary. And one measure that they looked at was biomass normalized primary production. So essentially the primary production rate normalized to the chlorophyll. And they plotted this against nutrient concentrations. And one thing that they found that was striking to them was this relationship where higher rates of production to biomass ratio were found when ammonium concentrations were below some threshold. And they set that threshold at around 10 micromoles of nitrogen per liter. And so they suggested that perhaps ammonium or some other contaminant associated with ammonium was leading to suppression in primary production. So this idea that phytoplankton respond differently to different forms of inorganic nitrogen is not new. It's something we've been studying for decades and decades. There's been several really nice reviews on that. Quay Dortch in 1990, and more recently, Pat Glibbert in 2016, and again in Limnology and Oceanography. One of the best well understood of the relationships or interactions between different forms of nitrogen is that ammonium concentrations, even at very low concentrations, will inhibit phytoplankton access to nitrate. And this is especially true for diatoms, which are generally thought of as nitrate specialists. So by way of setting this up and sort of teasing apart this somewhat counterintuitive idea, I did want to provide a couple of conceptual models. This again is from the Glibbert paper. And I ask you to focus your attention first on the right-hand side of this slide here. And what's put up here is two different scenarios, one in which ammonium makes up the dominant form of dissolved inorganic nitrogen. And in these systems, we would speculate that small cells, flagellates or picocyanobacteria, tend to dominate ecosystems that are flooded with ammonium, or ammonium at least is the dominant form of nitrogen, even at very low concentrations. The idea is these cells are small, so they have large surface area to volume ratio. They're really good at acquiring very low concentrations of scarce nutrients like ammonium in most of the surface ocean. And their nitrogen transporters to actively transport nitrogen across the cell wall is geared towards ammonium. On the right-hand side is the opposite scenario where you have a larger proportion of nitrate relative to ammonium. And in these ecosystems, we tend to see a dominance of diatoms. These are large, fast-growing cells. They are particularly well adapted to areas of like coastal upwelling or equatorial upwelling where you have large pulses of nitrate. And again, their transporters reflect nitrate assimilation. On the left-hand side is another way of thinking about this. This is now looking at uptake kinetics. So this is ammonium uptake versus ammonium concentration. And what this plot suggests is that as you increase the substrate, the amount of ammonium, uptake rate increases until it reaches some saturating concentration. We call that Vmax. What's interesting is that some studies that actually go into higher and higher concentrations of ammonium actually find that ammonium uptake starts to be inhibited by increasing concentrations of ammonium. A little bit further, what we find is that the transporters responsible for phytoplankton bringing ammonium into the cell, they are induced by low concentrations of ammonium. And the idea here is that they've evolved the ability to ramp up transporters when ammonium is scarce in the environment. They are able to scoop up the limiting resource. Nitrate behaves a little bit differently. Whoops, excuse me. Nitrate behaves a little bit differently. So here we have nitrate uptake versus nitrate concentration. And initially it too follows this classic, what we call Michaelis-Menten hyperbolic kinetics, where increasing concentration leads to substrate saturation. But sometimes we see a second phase of uptake, and sometimes it can even be a linear uptake. So nitrate continues to ramp up as substrate concentration goes up. And for nitrate transporters and enzymes like nitrate reductase, those are induced with increasing concentrations of nitrate. So higher concentrations of nitrate ramp up the ability to acquire nitrate. This has led some, including Dick Dugdale, who's been working on this in San Francisco Bay, to call ammonium sort of a paradoxical nutrient. On one side, we think of it as supporting lots of biomass and cultural eutrophication, low DO events, and harmful algal blooms. On the other side, in systems that are diatom driven, like San Francisco Bay and even the Delaware Bay, ammonium could actually lead to the suppression of fast-growing diatoms and therefore reduce overall production. Kind of an intriguing idea. So the goal of our study then was to use a combination of observational and experimental approaches to kind of revisit this question put forth by Kohei and John, this idea that ammonium or wastewater might be suppressing phytoplankton growth in the Delaware. And this little map here shows sort of our study region here, whoops, sorry about that. The thumbtacks are station locations across a transect. And then I put in red triangles to indicate where we did follow-up enclosure and kinetics experiments. I'm going to show you those in just a minute. Essentially upstream of the major wastewater inputs within the urban region, and then in the far field downstream at Newcastle. So our approach was to do a few different ways to tackle this problem. First is through a series of surveys occupying those thumbtack station locations. We primarily sampled in the summer, June and July. We had one spring cruise. And what we did was we characterized the dissolved inorganic nutrients. We looked at the amount of phytoplankton biomass. And we also used stable isotope tracers to measure primary production rates and nitrate and ammonium uptake. At the 3 stations in the triangles, we also collected 10-liter cubitaners, which we brought back to the lab in Lewis. And to those, we amended with nutrients. Either we had an unamended control, to some we added phosphorus additions, and to another set, we added effluent ammonium at concentrations that were above the thresholds that were suggestive of inhibition. Finally, we did some small bottle kinetics work where we made serial additions of either clean ammonium chloride or effluent ammonium, and then traced uptake of nitrate and ammonium to see if we could characterize the kinetics. So I'm going to share with you first results from the transect. This is dissolved inorganic nitrogen distribution along the river transects. Along the x-axis is river mile upstream on the right-hand side, starting in Florence, down to the left-hand side at Newcastle. With ammonium on the left, the center panel being nitrite, and the right panel is nitrate concentrations. The first thing I want to point out to you is that during our sampling period, generally we were below the 10 micromolar threshold suggested by Yoshiyama and Sharp as where we would anticipate some suppression to take place. I also put on here a 4 micromolar cutoff. This is what we have found experimentally in the San Francisco estuary as where we see strong ammonium suppression of nitrate uptake and through that reduced growth of diatoms. The center panel nitrite is just to remind folks that, that there is substantial ammonium inputs here within the water column. It appears that we're seeing nitrification, and nitrite is the intermediate form. So we're seeing microbial processing of ammonium, another important process. And then finally, the nitrate plot is just to remind us all that the Delaware has a very large reservoir of nitrate. However, if we are above a 4 micromolar threshold or somewhere in that area, much of that pool is not going to be used by phytoplankton, especially diatoms, and instead it's going to be advected downstream into Delaware Bay, where then it might fuel production. Here's the pattern for chlorophyll A. So here's the chlorophyll, again, same orientation upstream to downstream. We found 2 phytoplankton blooms in the summer of 2019. Otherwise, the chlorophyll concentrations were sitting between about 5 and 7 micrograms per liter. So these plots here are our attempt to kind of replicate the work of Yoshiyama and Sharp. And so on the left-hand side, what I've plotted here is the nitrate uptake rate normalized or divided by the particulate organic nitrogen versus ammonium concentration. And the take-home message from this, and we see this also in our enclosures, is that nitrate uptake does increase as ammonium concentrations go low. This is absolutely what we would anticipate from the literature because ammonium does inhibit or suppress phytoplankton nitrate uptake. The right-hand side is really analogous to the plot in Yoshiyama and Sharp. Here, it's carbon uptake normalized to particulate organic carbon rather than chlorophyll versus ammonium concentration. Again, we see this both in our enclosures. This is from the surveys. And again, higher carbon uptake per unit of carbon when ammonium concentrations are relatively low. Here's the rub though. When we go and look at the enclosures, these are enclosure experiments that we bring back to the lab. We amend them with effluent or phosphorus, so effluent ammonium or phosphorus additions. And then we essentially let a phytoplankton bloom progress through about 100 hours. What we find is there is virtually no difference in the trajectory of biomass production across the different treatments. We didn't replicate within experiments, but we did a whole bunch of these. So we replicated these through time and space. So at different station locations, upstream and downstream, as well as many different cruises, the pattern is very, very consistent. So the unamended control versus an effluent addition, effluent ammonium, we see the same trajectory. If anything, we see the effluent start to peel off and actually build more biomass late in the enclosure experiment. This is almost certainly because the unamended controls are running into nitrogen limitation. Finally, I want to share with you the results that are really probably the most intriguing to us, and these are the kinetics experiments. So here's
2 minutes, Alex, 2 minutes.
Oh, thank you. Startled me there. So this is ammonium uptake versus ammonium concentration. And the red open circles are when we add ammonium chloride, so clean ammonium salt. And what you can see here is that we were very reliably able to model classic Michaelis-Menten kinetics up to a very high concentration, 100 micromolar ammonium. So we see this saturating kinetics, and that is absolutely what we would have expected. The story is different though when we added effluent ammonium. So same experimental approach, we added increasing concentrations of ammonium, but this time coming from effluent sources. And here we saw very consistently inhibition kinetics. Sometimes the inhibition started at very low concentrations. Often we saw inhibition begin to kick in between 20 and 40 micromolar ammonium. And then we would start to see pretty strong inhibition of ammonium uptake. So to summarize, our results suggest that if we're seeing an effect, our evidence would strongly point to some inhibitory substance that is associated with ammonium and likely in effluent, much more likely than ammonium itself to suppress the primary production. Within the river in our surveys, it looks like the effluent ammonium load is being sufficiently diluted in situ to at least be below what we found to be experimentally to cause an impact. So it seems like in the river, we're probably below a threshold of concern, and we should be thinking about identifying any sort of contaminant. One last thing, which is a pretty big caveat, is that we do have still some work to do to understand if we're shifting phytoplankton community composition. So we, in our enclosures, we know we're growing chlorophyll. What that chlorophyll is still not very well characterized. We do know from drawdown of, say, nitrogen and silica that we are certainly supporting diatoms across all of our grow outs, but we do have some more work to do, and we have some pigment and other data to tease that apart a little bit more. So I think that is the end for me. I did want to acknowledge folks involved in the project, and hopefully I'll have an opportunity to answer some questions for you.
Yeah, so Alex, thank you so much. That is, as I said, not just intriguing, but now I know I'm full of questions, but unfortunately we need to keep moving. I hope that you can stay with us, for another 10-15 minutes in case we have time to get to questions. Our last talk is a lightning talk, so hopefully we do have some time. And so with no further delay, I want to introduce Chris Petrone from Delaware Sea Grant, and he is going to close us out in the hot topic session with his presentation titled Research and Outreach Rapid Response to the 2020 Broadkill Beach Oil Spill.
Great, thank you, Danielle. I'll try to blow through this, not too fast, but as fast as possible so that we don't go too far over and get some questions in. But thank you to PDE for the opportunity to speak today during this hot topic session. I'm going to provide a pretty quick and dirty overview of Delaware Sea Grant's response to the Broadkill oil spill back in the fall. So for those that aren't familiar or just don't remember, there was a small oil spill that came up on Broadkill Beach, Delaware. The first initial activation was October 19th, and Unified Command deemed the area cleared as of November 13th, 2020. The oil extended from Bowers Beach, Delaware, down to Assateague, Virginia. So you can see on that map there, about 87 miles or 140 kilometers long. And this was not a full tilt, beach covering oil spill, but definitely washed up in spots here and there throughout that range. According to federal law, the response is guided by the United States Coast Guard, and assisting them is the Delaware Department of Natural Resources and Environmental Control. And on the animal side is Tri-State Bird Rescue and Research Incorporated and the Marine Education Research and Rehabilitation Institute, who deals mostly in marine mammals. There were about 215 gallons or about 5 barrels spilled. That was the initial estimate early on in the spill, and then they carried that estimate through to the end. It was found to be high aromatic heating or diesel fuel oil, so not crude oil that was washed up. and about 85 tons of oil debris was cleared from the beaches throughout that time. I reached out to some contacts in local chambers of commerce to see if there was any sort of economic impact to the spill. Fortunately, there didn't seem to be any, despite Lewis Beach, Dewey Beach, and Bethany Beach all closing their beaches during the oil spill to avoid any contact with the oil. And the source, as of even today, is still unknown. So there's still an ongoing investigation on where that oil came from. So as you noticed, Delaware Sea Grant, the University of Delaware are not listed in first responders to oil spills. We have no business being out there in some respects. Let the Coast Guard and DNREC do their thing. So we looked to augment the cleanup any way we could. And so we talked about having an emergency response plan to oil, but we just never put one together just because it was such a low risk, perceived low risk here in Delaware. So we fell back to what we knew. And that number one was communications. So simply amplifying DNREC messaging, unified command messaging, however we could, whether that was through social media and some other avenues, which I'll talk about next. But so you can see through Facebook and Twitter, both personal and through the Sea Grant channels, we were able to amplify the messages of DNREC and really get the information out to those that follow us. We know education outreach very well. We had a few inquiries from classroom teachers that we work with a lot and for classroom presentations. So you can see me there on the left giving a presentation to a Cape and Lopin High School chemistry class about oil spills and oil fingerprinting and that kind of thing. And then on the right, hopefully you've heard of our 15-second science series, but we did the inaugural first time ever 7-day rapid response 15-second science series. So you can see the titles of the videos there. We were fortunate to already have one in our files from the oil spill response vessel Del River. So we threw that into the mix and we came out with this really nice 7-day series of 15-second sciences that was shared on all of our social media. We do have small pockets of money that we are able to deploy quickly. And so you can see on the left there, we initiated a rapid response research RFP and had a few inquiries. And we went with Dr. Art Trembanis from the University of Delaware, who did some side scan sonar work of about 160 acres off of Broadkill Beach to look for oil in the sediments. And so they ran their AUVs and a few other vessels to look for oil that had made it down into the sediments. And so they have lots of baseline data from this area already. And so that was a nice contrast to see if there was any. Fortunately, they did not find any oil in the sediment, which was a great thing. And then just facilitating connections with media. We had a lot of inquiries from all sorts of different media outlets. And so we were able to place different specialists, whether they be Sea Grant specialists or university faculty researchers, with media, newsprint, all sorts of news channels and things. And so we were able to act as that liaison to get connections to the media, which was pretty handy. So to sort of wrap up the lessons learned, we've heard this countless times from our colleagues in Sea Grant in terms of hurricanes, oil spills, tornadoes, floods. Make those connections with people that you may need or may need you before you actually need them, right? So make those connections. You shouldn't be meeting at the spill scene. You should make those connections well beforehand. Having a generalized emergency response plan. If you would have asked me back in January of 2020 if we would have needed a response plan to oil spill or a pandemic, I would have thought you were absolutely crazy. And so have that generalized response plan. It doesn't have to necessarily be specific, but what would you do in cases of X, Y, and Z emergencies? As I mentioned, we fell back to what we knew in terms of outreach and communications, and don't reinvent the wheel. So we extended and amplified not only DNREC messaging, but NOAA resources on oil spills. And then I'll show you the link on the next slide, but the Gulf of Mexico Sea Grant Oil Spill Science and Outreach Team has a tremendous amount of resources. And so we were able to amplify those messages and get that and disseminate those resources out to people that were interested. And then, with any sort of program, you always want to reflect and evaluate, make sure you did everything the best you could, what worked, what didn't, and make a plan if this happens again. So those are two links there. For more information, we have a new Delaware Sea Grant oil spill website, which includes that one pager you see on the right, all about the Broadkill spill. Our 15-second science episodes, a really nice timeline of all the major oil spills in the Delaware River and Bay, and then an oil spill frequently asked questions. And the link there to the Gulf of Mexico Sea Grant oil spill science team, just a wealth of information and resources for all sorts of different audiences, all different aspects of oil spills. Go check out that website. And with that, I can take questions or we can move on. So thank you very much.
Awesome, thanks, Chris. And I'd just like to add that after the Athos spoil, which was a big focus of our first summit in 2005, a federal advisory committee was formed, and a long story short, after a few years, a report was issued called the Delaware River and Basin Oil Spill Advisory Committee Report. It has all these recommendations. That's also available on PD's website, and I think from the US Coast Guard site. A lot of the same things that you just said in terms of recommendations and things we should be doing better. But thank you for that lightning talk. We do have, we're going to allow for 5 minutes of Q&A for all of our panelists, because we really didn't have time after each presentation. And then we'll close out the conference. But I believe, Kurt, are you there do we have any questions for any of our speakers in Hot Topics?
Yeah, so Alex, a couple for you first. There's a couple simple ones. So which wastewater treatment plants were the source of effluent? Obviously it was in Philly, but which one specifically?
Yeah, that's a great question, and perhaps somebody from PWE can answer that. I don't know if they're muted or what. We had a single composite that we were given at the beginning of the study within the sample region, and I forget the name of the plant. I should have known that. We did ask for a 24-hour composite sample, so it's sampling through a full day's worth of discharge. So we were attempting to get an integrated sample. We then analyzed that for concentration and then use that as in a dilution series. So I'm sorry, I don't have more specifics about the effluent.
Gotcha. So you might not be able to answer this, but do you know if PWD disinfects its effluent? And if anyone from PWD is on, feel free to raise your hand and we can unmute you.
Yeah, again, I would a little bit outside of my expertise. And of course, our next thing, we're finalizing our report here. And part of that, sort of next steps questions is to sort of move to identifying potential contaminants, not my area of expertise in toxicity, but something like a TIE set of experiments that could be done. In San Francisco Bay, we do think a lot about disinfection byproducts, chlorination, dechlorination, and those sorts of things.
All right, and then a question about light availability. Do you think light availability plays a role in ammonium versus nitrate uptake?
Absolutely. Light is important in all of this. And so in our enclosure experiments and survey, all of our incubations, we eliminated light limitation. So we provided them with saturating irradiance in all cases. What we do know is that typically ammonium is less light limited in terms of a process. Nitrate uptake is much more strongly governed by light limitation. So you see a curve much more similar to what you'd expect for carbon assimilation by algae for nitrate. And so those are important interactions in areas that we're certainly actively working.
And just real quick, we have a note from Joanna, your co-author, Alex, that the effluent sample was from the Southwest Water Pollution Control Plant.
Thanks, Joanna, for bailing me out.
Kurt.
I had a quick question for Dave, actually. Did you talk to any of the producers, either wild harvest or aquaculture? Did they get a sense of their shipments, whether there was any kind of shift in preference from raw to cooked, or was it still mostly raw? Because my understanding is raws dominates the market, but due to COVID, maybe more people wanted cooked oysters. I don't know.
I don't know the answer to that.
Kurt, do we have other questions? Right.
Nothing else.
I have a quick question for Adrianna. Adrianna, hi.
Hey.
So I'm curious about the, for the plots at Fortescue, what were the final, did you reach your expected target elevations? And I guess where did the plants end up relative to baseline in terms of where they were in their optimal growing ranges? Or if you want to think of elevation capital, did you actually buy elevation capital?
I think so. The way that the way placement happened at Fortescue, it was a little bit like, well, my son throws wet mud. So I think it was a lot like that, plops of material here and there. And so I think when you look across the whole site, it's the one site where we don't have nice, smooth gradients as far as elevation gain. And so when we play the game of averages, we lose a lot of that. So I would recommend, so at the plot level, we have elevations, and that's some analysis that we still need to do at the plot level, take the RTK point at the plot and compare it to the growth range, and then look at the species composition in the plot. The nice thing about doing taking the data collection at that level is we can answer those questions because we have that time series. But I don't know, I think the variability probably muddles that for now.
Great. Yeah, thanks.
I think Fortescue came in a little low, is my sense, my overall sense.
Thank you. Look forward to seeing more of the data. I actually had a question for Alex too.
One more, Danielle.
One more, okay. Sue Killam, who many of you know, who sort of helped found this summit, and the license plate on her car reads diatoms, a vanity plate. And Sue always talked about the phytoplankton dynamics with Jonathan Sharp and others in the Delaware. And I'm just curious with the ammonium story, she often talked about the silica or the diatom depletion relative to historic conditions in the system. And she was always curious about whether silica limitation. We've also, I think, developed almost a paradigm that the Delaware's turbid and there is light limitation. And now, obviously there's this concern about something related to ammonium, effluent ammonium. So I'm just curious if you had any comments about whether silica might be limiting those diatoms, or maybe Sue should have been looking more at ammonium rather than silica.
Sorry about that, I was muted. First thing is, the system is strongly light limited until you get down into the lower part of the estuary. And forgive me, because it's been a long time since I did my dissertation in the Delaware, quite a long time ago. But I guess often we think about, or I thought about it, it's almost like a chemostat where you had nutrients coming down and largely being ejected down into the lower bay, where it then is mixing with a much larger light field. And that's when you start to see sort of mid-estuary large diatom blooms in the spring, and then biomass increase in the summer, late summer. But I'm not certain if that was really diatom related. Somebody probably would remember that better than me. Silica limitation is certainly a potential limitation for diatoms, and a really simple thing to do is just to look at the stoichiometry of the nitrogen concentration. If you use something like Mark Brzezinski's 1-to-1 ratio for diatoms, DIN to silica drawdown ratio of 1-to-1 would give you some predictive ability about where limitation would take place. Chances are it would be in the lower part of the estuary, though, where the light field begins to open up.
Thank you. Okay, I'm getting a signal from other folks. Listen, hot topics has always, been a hallmark of the meeting. I want to thank our 4 presenters for upholding that tradition. So I'm going to do a little round of applause because we're actually going to transition right into the close of the conference. But for right now, for hot topics, yay, imagine lots of noise and applause. And okay, Sarah, go ahead.